And welcome to section 4.8. It continues where we are going to talk about the function of the nucleus and the ribosomes. So cellular activities are going to be dependent upon protein synthesis. That is the overall picture here, is that we need to create so many proteins, and it is directed by DNA. We've got these two processes here that are so vital to this protein synthesis process. First, we have transcription, and in transcription we are going to use ribonucleic acid, or RNA, and a copy of a gene formed from DNA in the nucleus. So you see over here in which we are going to have this take place in the nucleus, and a mRNA strand gets created and is transcribed from our DNA molecule. It is then going to leave the nucleus and enter into the cytosol, and we are going to start the process of translation. We're going to use RNA for synthesis of the protein using our ribosomes here, right within the cytosol. So you can see the ribosome here is reading the mRNA strand and using tRNA to come in and add these amino acids to build the protein. Remember that amino acids are the building blocks of proteins. So here are some of our required structures. We already know that DNA is going to be a major structure needed in transcription. It's going to serve as our template for that complementary RNA molecule. So just like we saw over here, this DNA molecule that was twisted into a double helix gets opened up like a zipper, and we create this mRNA strand within it. We see repeating ribonucleotide monomers, and remember, when we have RNA, that thymine gets changed out for uracil. So we don't have thymine within an RNA strand. We replace the T's, the thymine, with uracil. And each of the sugar molecules is not going to be deoxyribose, it's just going to be a ribose sugar now. We've got phosphate, and then we can add one of those 4 nitrogenous bases. And we aren't going to have RNA as a double helix. Instead, it's going to be a single strand of nucleotides. We'll also require large numbers of ribonucleotides and an enzyme RNA polymerase. Again, this is going to be an enzyme. And this is located in the nucleoplasm in the nucleus, because we know transcription takes place in the nucleus. And we have 3 types of RNA that we are going to produce during transcription. We've got our messenger RNA that we've talked about. Here's our backbone with the ribose and the phosphate, and then here are the nitrogenous bases A, U, C, and G. And then we've got our rRNA, or ribosomal RNA, which is going to help read the messenger RNA to create the proteins. And then we've got our transfer RNA, or tRNA, that would have the anticodon over here that would match up to messenger RNA and the attached amino acid. So there are 3 events in transcription. They are initiation, elongation, and termination. So let's take a look at how this process takes place. With initiation, we are going to see that DNA is going to become unwound by specific enzymes to make it accessible to RNA polymerase, which is pictured in the purple over here. The enzyme is going to catalyze the synthesis of mRNA. And RNA is going to attach to the DNA strand here and locate the promoter region. So this promoter region really helps create a start point for that mRNA strand to start. Bonds are going to be broken between DNA and the region will expand in order for this to happen. Our DNA strand is going to be copied, and we will have that serve as our template strand. The other strand will be our coding strand that is not copied. You can see that coding strand over here. Now let's talk about the second step in transcription, elongation. Our free ribonucleotides are base paired, and so our pair with exposed bases on the DNA template strand is going to be red, and we get formation of hydrogen bonds, which you could see pictured over here, between the ribonucleotide and the complementary DNA base. So notice how here on the DNA we have thymine, but it is going to be paired with adenine over here on RNA. And we have adenine over here on DNA, and that is going to be paired with uracil on the RNA strand. And we see a new phosphodiester bond between the ribonucleotides, and this forms our RNA polymer. Our enzyme RNA polymerase just moves down the DNA strand and continues creating our mRNA strand until the entire gene is transcribed. And our last step is termination. So RNA polymerase will reach that stop code at the terminal region and release the DNA strand and mRNA strand. We'll see that the hydrogen bonds will break between the DNA and the new mRNA strand, and DNA will rewind up into its double helix. So what we're going to do now is watch an animation of how this is all taking place.
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. When RNA polymerase reaches a specific sequence of nucleotides on the DNA, called the transcription terminator, a hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
So next, before we move on to translation, we have to make some modifications of our mRNA. So we really should call this pre-mRNA, and we're going to make these changes before we leave the nucleus in order to form our mature mRNA. It's used as a recipe in order to make the protein. So we're going to use something called splicing. The non-coding regions of our pre-mRNA strand are called introns. So they're going to be removed from this pre-mRNA strand, and the exons are going to be transcribed nucleotide sequences in our mature mRNA. So we splice them, and notice how the intron regions are no longer existing in this mature mRNA strand. Subsequently spliced together by something called a spliceosome. So here is what that spliceosome looks like. You can see our pre-mRNA is moving through, and the exon regions are being kept, whereas the intron regions are being sliced out in order to get our mature mRNA strand. We're going to see that the pattern of splicing does vary, and it provides a means of producing a larger number of proteins from DNA. Some other changes that take place to form our mature mRNA is the bonding of our ribonucleotide containing guanine. So we're going to go through a process called capping. will bond the lead end of the mRNA strand, and this will help prevent digestion by enzymes within the cytoplasm once it reaches there. And then we have an addition of a poly A tail. We are going to remove the terminal segments of our mRNA strand and replace it with the adenine nucleotides at the tail end. And you can see multiple mRNA transcripts are possible. And at this point, our mature mRNA is going to exit the nucleus following these modifications. And we are going to start the process of translation in order to synthesize our protein. It's going to occur at those ribosomes that we talked about within the cytoplasm, and our mRNA strand is threaded through the ribosome. We are going to look at the nucleotide sequence on our mRNA strand, and that will translate into an amino acid. So I want to show you something really quick that you are going to do on your lab or within your lab manual. So this is a part of, part 4 in your lab manual. And what you're going to see is this chart right over here. So if I flip back to our PowerPoint, I know it's a little off, you could see that there are these codons or codes in which we're going to take 3 of these nucleotides and they will translate to an amino acid. So for instance, if I read GUA, I'm going to refer back to this chart over here and start in the center, GUA, and I get valine. And that's what I had over here as my amino acid. Let's do the next one. I have UGC. So I'm going to start in the center, U, G in the second layer, C, and I get cysteine as my amino acid, and that's what CYS is representing, is a cysteine amino acid. So on your lab manual, I'm going to give you a pattern here of our mRNA strand, and you are going to write in your amino acids using this chart. So again, you could do that on Blackboard. So let's move on to talk about our ribosomes. We need our ribosomes in order to create these proteins. It really reads the codes in order for us to know what protein to, what amino acid to attach. So our ribosomes are composed of rRNA and protein. We have that large subunit that has the A site, P site, and the E site, and then the small subunit here. And these rRNAs serve as catalysts during the assembly of these amino acids into proteins. If we look at our mRNA strand, we got this from our genes. It's carrying instructions for the proteins, and we see that it has a linear sequence of nucleotides. So again, we take 3 of these nucleotides in order to code for a specific amino acid. So AUG is our start codon. That's going to tell our ribosome, hey, start over here and start building, and it'll read each of these codes and attach amino acids. And that's basically what this says over here. So we've got our start codon signaling protein synthesis, and then we're going to read through until our stop codon. And down here we have our tRNA. This is going to bring the specific amino acid to that specific mRNA codon. It has this clover leaf shape to it. So while this looks a little bit complex, most of texts and animations will have this T-shaped blob representing your tRNA. And this is going to plop down into the ribosome in order to attach the specific amino acid. So your tRNA is going to contain what's called an anticodon, meaning whatever we are reading here. So let's just use this example of AUG as the start codon. The anticodon is going to carry the opposite of that. So I shouldn't say opposite, but the pairing to it. So for A, we're going to put U. For U, we're going to put A. And for G, we're going to put C, so that the anticodon matches to our codon on our mRNA strand. And over here, we have one of those tables to give you the name of the amino acid. So I know this is really small, but it is pictured in your book. It's just a different way of figuring out the amino acids. So while I showed you in your lab manual, I had that circle diagram to do it. This is more of a linear chart. So the first letter would be here. So let's just use our start codon, AUG. So there's our A. The second letter is U. And then we would look down here for G. And this is really small. So there's AUG, and methionine is our start amino acid. So that would be the first amino acid that is created. And this is all catalyzed by an enzyme known as aminoacyl tRNA synthase. After binding of an amino, that tRNA is a charged tRNA. It's going to serve as an adapter site for binding the tRNA to the complementary codon of mRNA. And also we have 20 amino acids here found in proteins of living things. So I wanted to show you this figure as well. This is showing you the structure of an amino acid. You could see a carbon in the middle. We've got our hydrogen. The R is a variable region. So this is going to change depending on the amino acid. We've got an amine group here and a carboxylic acid group over here. To represent these amino acids simply, we're going to use a sphere. And this is showing us a strand of amino acids. So those tRNAs place each one of these amino acids in order to make this chain. What happens is this is a primary structure of a protein, and then we start moving into higher levels of structure. So we have a secondary structure in which we get some folding, a tertiary structure which we form some other bonds, and finally a quaternary structure where we get our full protein, and we have many strands of these amino acids placed in here. So if we're starting small, we start with amino acids. Lots of strands of amino acids attached to each other create peptides, and lots of peptides create proteins. And here's that full chart, a little bit easier to see here in case you wanted to use it. Now for translation, we've got 3 events again: initiation, elongation, and termination. So let's start with initiation. This is going to be a complex formed between the ribosomal subunits, our mRNA strand, and tRNA. We've got our base pairing between the tRNA and our start, that should say codon, not colon, in our mRNA. And methionine, we said, was the first one. AUG is what we code for. So you can see that our tRNA has methionine attached here to start our protein synthesis. Later, this can be removed if it's not needed. And notice how we are in the P site. Now for elongation, we're going to get delivery of each of these amino acids by specific tRNAs in order to form our protein. So we've got 3 steps. Our charged tRNA with the complementary anticodon base pairs with the codon of our mRNA comes to the A site. We get a peptide bond, I guess this is pictured over here, a peptide bond that is formed between the amino acid, my mouse keeps disappearing, in the P site to the amino acid in the A site. And then our ribosome is going to move it 1 codon downstream. So basically, this tRNA in the P site is going to move to the E site, and this tRNA in the A site is going to move to the P site. So once that A site opens up, another tRNA strand moves into the A site, And we're going to have that peptide bond now form between this amino acid and this one. And in the E site, the tRNA strand is now uncharged, so it gets released from the E site. So you could think E for exit. See you on out. And we're going to repeat this over and over again until the mRNA strand is completely translated and we hit that stop codon. And as a result, we're going to get a chain of amino acids building up top here. So we've reached termination. We've got the entrance of our stop codon, and that has entered into our A site here. So we're going to stop and end translation. Our release factor enters the A site instead of a charged tRNA. At this point, our ribosome subunits are going to separate and the new protein is released so that it can go on to its folding processes to become a complex folded protein. Now we can have polyribosomes. This is when our mRNA has many ribosomes attached to it. So here is an example where we have our mRNA and lots of ribosomes all around it. And in this way, we can make many copies of a protein quickly. Now let's revisit DNA. This is going to be our control center of the cell. Our DNA directs synthesis of our proteins that are carrying out our body functions. It's indirectly responsible for other metabolic changes, the making of our steroids, the enzymatic pathways of glucose oxidation, and controls the enzymes responsible for things like our decomposition and synthesis chemical reactions.